So welcome to the PHOENIX webinar, a new player bridging nanopharmaceutical innovation from design to clinic. PHOENIX is an open innovation testbed for nanopharmaceutical innovative products. You can find our website here. It's PHOENIX-OITB.eu. And we'll have all our contact information at the end. You'll see we have a couple of short presentations at the beginning. And then we'll have a very short break and then a round table with question and answer. So again, most of the questions will be taken then. If you have any questions for the first presentations, the general ones, Professor Khinast has to leave at 10:30, so we'll take questions for him first, and then the rest will come after the round table. All right, then I will hand over to our first speaker. We're going to start with our project coordinator, Tommaso Serchi, from the Luxembourg Institute of Science and Technology. He's going to give us a brief overview of the project.
Thank you so much, Caitlin, and thank you everybody who is online today for coming here. It's our big pleasure to show you what we do in PHOENIX, and most importantly, why we do it. So PHOENIX is a European project supported by a European project. The extended name, you can see it on the screen, is Pharmaceutical Open Innovation Testbed for Enabling Nano-Pharmaceutical Innovative Products. It was conceptualized to answer the specific challenges of this project here, of this call, which deals with open innovation testbeds, and specifically for nanopharmaceuticals. So open innovation testbeds are projects which are meant to provide tangible services to the market in order to foster innovation, targeting a European market and European industry, and particularly small medium enterprises. Now, this is particularly important in nanopharmaceuticals, which is probably a bit special field compared to other fields. It's still a niche. It is very promising. There are a lot of thriving SMEs out there doing innovation. The problem is that the process is very, very, very complex. So there is a strong potential for better therapies, more effective therapies, but they somehow don't manage to go to the market. And there is a major challenge that normally this kind of innovative small companies do not have the capacity to go to GMP production, to produce their products in sufficient quantity, and normally they don't reach the preclinical and clinical testing. So there are some requisites that you need to have in order to be successful. So the process, first of all, from the conceptualization to the clinical studies and full exploitation of the product, it is very, very long. You see here there is a timeline which is indicative. It can be, of course, be shorter, but most probably it will be much longer. And it takes several years, which also means that at the very beginning you have a high economic risk, a high risk, and the expenditure are not so much in the very beginning. They rapidly go very, very high as you proceed towards innovation, of course, reducing a little bit the risk because you have made choices in order to reduce the risk. In order to make all this process more sustainable, there are some requisites. First of all, we need to provide to the market affordable and advanced testing, manufacturing facilities, because we cannot imagine that every small company will set up their own GMP facilities. And then this is followed by a number of services which are possibly existing, but they are very fractured and not easily accessible, and definitely not at low prices. And all these problems bring to the difficulties that are going from a lab bench to industrial scale production. It is extremely challenging, both economically and technically, and this hinder a lot the entering of the clinical trials. To answer all these questions and challenges, we had set up the PHOENIX consortium. These are the numbers in a nutshell. We are in total 12 partners from all across Europe, and you see the names of the institution and the logos here on the slide. It's a project which started back in 2021, and it will last until February 2025. It's not a small project. The total project cost is slightly short of 15 million. of which 11 million are contributed by the European Commission. Which also means that there is a large investment from our industrial partners who are not fully refunded, and that's the discrepancy. We're showing also the interest that there is from industrial realities across Europe to develop the sector. And the idea was to put together a strong consortium to develop services starting from the very, very beginning and give them to the market as long as the project progresses. This is the geographical distribution from Europe. As you can see, we are quite spread. And I can tell you it's a very nice consortium to work with. And let me just finish the... So what it is the objective in a nutshell of PHOENIX? So what we want to do is to try to provide solution to overcome the valley of death of innovation. So it is a common concept in all innovation. Some sectors are a bit faster, a bit less risky. Pharmaceutical is definitely risky, nano-pharmaceutical even more. So what we want to do is to enable the seamless, timely, and cost-friendly transfer of nano-pharmaceuticals at any of their stage of development, so be it conceptualization, lab production, whatsoever, to the market and make it affordable. So we want to foster the innovation in Europe by providing services which can be used really from SMEs and timely in where they need to be used. Now, as I said, what we want to do is to enable a smooth transfer from lab scale to GMP grade. And this implies a lot of different services and knowledge. Thus, what we want to create is an ecosystem of different entities providing different services at different expertise, so that we can cover the whole production line. Now, the idea is to create, and that has been created, the PHOENIX SEP, single entry point. It's a legal entity which will be the one taking the providing the follow-up of the project and providing access to the services. So the SEP is conceptualized as being a one-stop shop where customers, doesn't matter how big, doesn't matter what they need, can enter and leave whenever they want. So the idea is you need regulatory support, you can go in, you get the services you need, you go out. You need research and development, you go in here and then you go out. Or if you need everything, you enter and then you follow the line across the different services. This will allow access to manufacturing facilities. We hope, and we are pretty sure, that we can ensure a reduction of cost for production and also support and reduction of cost for regulatory compliance, harmonized condition for quality assurance, scaling up GMP production, but also help in the conceptualization of a product if it is needed at the very beginning, or support for the commercialization and industrial implementation at the very end if that is needed. These are the macro categories of PHOENIX services that we have developed, and they cover innovation aspects, manufacturing, characterization, considering physical, chemical, in vitro toxicity, preclinical in vitro toxicity testing, and preclinical in vivo characterization using animal models. And of course, in all of these, we always use state-of-art services, and as much as possible, we use officially approved guidelines. So I hope that in this very short overview, and I hope I wasn't over time, we have shown you what we do to try to improve the European infrastructure and overall the competencies in the nanopharmaceutical process development, including characterization, testing, control, and production. And this, we hope, in the future will increase the attractiveness of Europe as a location of choice to carry out advanced medical and nanopharmaceutical products. I didn't mention, but one thing that we have, which differentiates us a lot from competitors, is that we are used to work on tailor-made solutions. So there is basically nothing that, given enough time, we can support developing. And then, in turn, we hope that all of these will improve the general supply capacity and availability of services producer and create the whole network and the whole ecosystem that we need to progress the nanopharmaceutical field. And this was my last slide. So as far as I understood, I stop now, and then I'm happy to take any question whenever it is the moment to take questions.
Great, thank you so much, Tommaso, for giving us this great overview of our project. And again, he will be joining our roundtable, so you can already write questions in the question and answer function or save them for the next part. Next, we're going to move to Johannes Khinast. He's CEO of RCPE. This is the Research Center for Pharmaceutical Engineering based in Graz, Austria, just out of a very successful conference on continuous manufacturing. So Johannes, yes. The floor is yours.
Thanks, Kathleen. Thanks, Tommaso. Can you hear me fine?
Yes, perfectly.
Yes, wonderful. So thanks also from my side. I'm also very happy to be part of this meeting. I think originally we planned to have this meeting physically in Graz, Austria, but whoever decided not to do this is a genius, because it's raining, it's 6 degrees, it's awful weather, so it's much better at home. And that allows you also to take a little more careful look at what we're doing here at RCPE. RCPE is a research center for pharmaceutical engineering. So our mission is to allow companies to produce, to manufacture their products, and especially I'm talking about innovative, novel products at a very high quality, at reduced costs, with very robust processes. And continuous manufacturing has proven to be one technology branch, one technology that allows you exactly to do this, to produce products at a high quality, at relatively low costs compared to other processes. And this is what I want to talk a little bit about today. And I also want to thank all my collaborators who actually have been the ones who have been doing the work. Let me start by saying that we are increasingly seeing that we have an international supply chain crisis. There are lots of medications where we have shortages. Many medications cannot be delivered to patients, and this is true for cancer patients. It's true for neurological diseases, for infectious diseases, HIV, bacterial infections, and even more, this is true for structured systems. So nanomanufacturing is still an area which is still in its infancy, and we need to do a lot of work to make the supply chain of nanopharmaceuticals more robust. So this is one of the missions, and advanced pharmaceutical manufacturing in many ways is a way of doing this. So because we need to produce those more complex products, nanopharmaceuticals are more complex. They combine multiple materials. They're structured on a nano level, and this structure is extremely important. So you cannot just put those materials together. So you have to make sure that the structure is important. When, for example, when we talk about mRNA nanoparticles, we need to make sure that the particles are intact and that the loading of the particles with the RNA is also efficient. So the structure is very important. It's more complex. We have a lot of variability in the process, but with advanced processing methods, we can improve or reduce this variability and therefore improve quality. That also allows us to shorten supply chains, which immediately leads to an increase in availability of critical medicines. It enhances resilience. We are here in Europe, and we are completely dependent on China and other Far East countries in terms of our medicine supply. It allows us also to have greener processes. I will show you this in a minute, why it is greener and why this is actually important. And it's also cheaper. I mean, cheap, it doesn't sound good, but if you have medicines which are very costly, right, and the reimbursement offices of the health providers don't allow you to use it, there is it's not a good idea, because then you cannot develop anything innovative. Because if nobody pays for it or buys it, then you have done this whole work for nothing. So cheaper is important. It's also important with respect to Far East producers, which can, of course, produce at much lower cost. So we need, that's what my conviction is, we need in Europe better technology, faster technology, and cheaper and greener technology in order to compete with other regions in the world. And this is especially true for nanopharmaceuticals, which are such complex things. I mean, I don't want to go too much into this idea of advanced pharmaceutical manufacturing, but being controlled, being understood, having an ability to model those systems, have a robust process which doesn't run out of its specifications if there's a small change in the inlet parameters. All of this is increasingly important. And here, as I already mentioned initially in my presentation, continuous manufacturing is, in my view, the key technology to make this happening. And I've been, how can I put this? I've been in the area of continuous manufacturing right from the beginning. At Rutgers University, we started doing continuous manufacturing. Companies were convinced that this is complete BS, so this will never work. But in the last 25 years, a lot of change of minds has happened. The FDA has been pushing continuous manufacturing. FDA is very much for advanced manufacturing processes. IMROH is also starting to realize this. Even Japan and China, companies and regulators are starting to look at this. And this is one of the books. I mean, I'm not getting a single euro for this if you buy it, but I just wanted to show you that we very early on started to actually develop textbooks. And these textbooks, the early ones, were mainly focused on solid dosage forms, meaning capsules, meaning tablets, meaning lozenges. All of this we have been working on. But now, in the last years, we started to apply this idea of continuous manufacturing also to nanopharmaceuticals. And I will show you a little bit what we've done later on in my talk. And that's also why it's exciting to work together with the PHOENIX partners, because they having the products, but we have maybe the processes, and we have maybe the process control, and we also have the modeling tools. So why is continuous better? It is better because to the left, you see a classical batch process. You have one step, then you stop, then you do a quality assessment. Then you go to the next process. You do the same. You stop, go into quality assessment. This goes step for step for step. So it takes you a long time. Plus, this is something which people don't realize. A batch process is always unsteady in its process characteristics, meaning you're starting with a high concentration and then you're going down the low concentration, or you're starting with a low concentration going up to a high concentration. So there's always a time-dependent trajectory, and this leads to huge variability because not every process is the same at all the times. So once you start maybe with 1%, and then you go to 95%. Sometimes you start with 1%, but you're only going to 91%. And 3 weeks from now, you're going to 85%. So there's huge variability due to this inherent time dependence of batch processes, which is not necessarily a bad thing always, but sometimes it's a huge problem because it leads to variability. Continuous manufacturing, in contrast, is a system where everything flows to a plant. Okay, it flows through a plant, and it is always in a steady state. So ideally, a continuous manufacturing process, once you started it, runs forever without any time changes. And this is the reason why there's much less variability, because it's running at a steady state. You don't have this turn on and the shutdown processes. You don't have these dynamics of a batch process, and you have a continuous process where nothing changes over time. This is the idea, okay? And that's why we have much lower variability and higher quality. And there's another advantage. If you have a process that runs 24 hours, 7 days a week, okay, and ideally a few weeks, you can do this whole thing in a very small setup, right? You don't need huge production halls with big mixers like you use in batch processing. You have a small system, you just let it run for a long time. And therefore, by using this, you can produce a lot of material in much smaller facilities, which is another major advantage. And here comes the third advantage. You can actually run the process on the same equipment that you developed the process on. Because typically, when you look at this left area, the batch paradigm, you have the small process, the kilo lab. Even before the kilo lab, you have very small, maybe gram labs. And then you're scaling it up, and this scale up is a pain. There's always problems, and there's always something not working, because scale up is something which is really badly understood, because there's so many processes which have different dependencies on scales. So scale up is always a pain. Therefore, continuous manufacturing allows you to run the same process on the development system. It's just running longer in order to produce industrial capacities. And that's the idea. So technology is there. I already explained it. It leads usually the massive speed up of production up to 20 times, going sometimes from half a year down to a few days. The plant size is much smaller. It's controlled because you need inline process control because it needs to be in a steady state, and you can produce on demand. It's agile, it's flexible. You can decide to run for a week. You can decide to run for a month, depending on what you need to produce. And that's, I think, the idea. And this applies to small molecule oral dosage forms, to all the synthesis, to drying, to the solid dosage form production from feeding, milling, blending, coating. That's where we started. We've been now also working together with others on biologics, so large molecules in bioreactors, which then are continuously purified, polished, filled and finished. So it works also for biologics, and for nanostructure systems, it works exceptionally well. And this is exactly what I want to show you today. So continuous processes for nanotechnology. And we all know that there's a huge variety of different particle production processes for nanostructures. Not all of them lend themselves easily to continuous manufacturing. I have to admit this, but many of those, without going into much detail, which ones are better and which ones are worse, many of those can actually be implemented in continuous manufacturing. And some of those we've been doing top-down approaches, for example, high-pressure homogenization, where you break down large particles to make smaller ones, or bottom-up approaches like impinging jet mixing, where you have two phases which are immiscible, but if you mix them in the right, correct way, then you can produce nanostructured systems. So top-down approaches and bottom-up approaches both can be used to make nanostructured systems. The important is just when you do the scale-up, that you maintain the characteristics of the small-scale development process to the large-scale process. That's exactly the challenge, and that's the reason why continuous manufacturing is so ideally suited, because you don't have to do this. You don't have to do a scale-up. There are many challenges, of course. You need quality control. You need to make sure that efficacy, safety is given, so that you have a reproducible process. All of this is important. And what we've been doing, I'm just rushing through this a little bit because if you're interested in more detail, I'm happy to send you publications, or you can contact me directly. We've been in the first phase now mainly working on nanostructured lipid carriers and solid lipid nanoparticles. So this is the area where we've been doing work, and just one line which has been realized as a demonstrator, kind of is a bottom-up line where we have two pumps. It's a very simple system. Pump one with organic phase, aqueous phase, which contains an API, and then we mix them in impinging jet mixers, which for our partner Knauer, there's a quenching line, and that's essentially, after solvent removal, you can have a continuous manufacturing of your product. And what you need, that's what I maybe didn't emphasize so much before, for continuous manufacturing, you always need inline process control. Okay, so you need to make sure that your process is at the steady state you want it to be. And this is only possible, it comes from the chemical engineering tradition of 100 years. You need online sensors, and these online sensors allow you to make sure that you are at the set point that you want to be at. So you need inline particle characterization, particle size, dispersity, loading. All of this has to be done in line. This is a little bit the pain of continuous manufacturing, because you need those sensors, but it's also a major advantage because you don't need the quality lab where you can do the release. You can do a release of your product purely based on inline real-time process data. And what we've done here, we've been using a few technologies. Just one I want to show you. This is the spatially resolved DLS. I'm not sure how much you're familiar with DLS. Essentially, simply speaking, small particles, they are prone to random motions, right? This Brownian motion, and this speed of the motion is proportional to the size. So if you measure the motion of these particles, then you can calculate the size. That's the basic, very simplified approach of DLS. Now the problem is when you have an online continuous flow system, the thing flows through the reactor, right? So now you have two types of motion. You have like a parabolic velocity profile, because in a pipe there's a parabolic velocity profile, so particles that the wall are moving slower than in the center, and superimposed is the Brownian motion. If you now use a traditional DLS, these two speeds of the particles are mixed together. You cannot resolve those. But if you do a spatially resolved one, you can understand why particles at the wall are moving slower, yet still show the same Brownian motion and a different speed at the ones at the center. So by doing so, you can use this system actually to measure particle properties in flow. And we've done this. You can see here, these are the particle sizes for different types of impinging jet mixers. These are 5 different impinging jet mixers. We have been characterizing them in real time. You can see very nice results you get here for different impinging jet mixers. This inline process monitoring works very well. Now we use this, of course, also not only to do quality control, but also to control the process, meaning we're using the sensor. If you see this here, this red thing at the real-time measurement in process LSB, we use this to control the process, meaning we control pump speed, loading, also the turbulence in the impinging jet mixer to get a product of the desired quality. We do a lot of modeling without going through this. Just want to show you one thing. This is, I hope it works now. Yes. So these are different set points. You can choose different set points, and then your D50 stays at the set point, right? And then you change the set point again, and it goes down to a new set point. So should be 102, then 116, and 128. So that's how you can control your process, advanced process control, and you can produce the particles of a desired property. We've also done this, and now I have to speed up a little bit because otherwise I'm late for my other meeting. We've done this for top-down production. This was bottom-up so far. This is top-down, so meaning high impingement jet, so that you break particles at due to high turbulence. Let me just show you. It's like essentially a system where you have a high pressure homogenization step, and you can let the particles go through many cycles, and you can see after a few cycles already you have particle sizes which are very robust in the area of 100 nanometers. Also the polydispersity index very good, having a quality definitely below 20%, so which is beneficial. And then the last part maybe want to say is we've been doing a lot of work in modeling, and modeling is extremely painful because the mixers or the systems are in a kind of a millimeter to centimeter scale. The molecules are much smaller, you know, like the nanometer scale. And what happens actually where the interesting stuff happens is the mesoscale, right? So the mesoscale is influenced by the macro scale, but also by the molecular scale. And modeling and simulating all of this is increasingly actually impossible. Let's call it impossible. And to this day, it's impossible. So you need information from micro scale. We need information from the macro scale. And we bring this all together to do simulations. This is shown here, the microscale nanoparticle, the molecules, the macroscale, that's the mixer, bringing together the mesoscale. And for this, we developed, I mean, some colleagues, not I personally, okay, one of our very talented computational scientists is working on these methods. It's called a boundary method. You can see here the very complicated process of breakup of a droplet. You can model this now. We can also model, let me just show you, like just ethanol, water. Okay, sorry, and apparently you can see here, we can model all this now. Let's see if you can see this, this impingement jet mixes, and can use this information now to understand how particles, nanoparticles are forming, how they're loaded, what's the quality, what's the polydispersity index, how process parameters impact the quality. All of this can now be done. We're not completely finished with this. So this is something which we'll need to do over the next years and refine and work and continue to work. But we started, and there are good results already. So in summary, we demonstrated continuous processes for nanoparticle formation, both bottom up and top down. We have a whole pilot plant. We have inline process control realized. We use it for process control. We have simulation framework developed. And we have a lot of expertise. So if you want to work with us in characterization, process development, quality assurance, also in terms of regulatory aspects, we are here for you. I want to thank one thing I just want to point out, we have a EDX course on continuous manufacturing. It's free, self-paced. If anybody wants to learn about it, then we can do it easily. And with this, I'm already done, and I hope it was partially interesting. Hope it wasn't too much information, just a very high bird view overview of what we've seen. Thanks, everybody. And with this, I'm done.
Great. Thank you so much, Johannes, for this presentation, and especially squeezing it in, a lot of information in a short time. So thank you so much for coming. Unfortunately, Johannes has to leave us, but if you do have questions, please still write them in the chat. We will collect them and send them on, as he said. Great. All right. Thank you so much for coming.
I'll be joining you maybe in 1 hour, okay? So see you later. Thanks, everybody.
Thank you. Now we're going to move on to our services. These will be presented by Jesus Martinez de la Fuente. He's at the Spanish National Research Council in Zaragoza. He'll be going over the services, just an overview, and then after a short break, we'll have our roundtable where we'll go into detail about these services with question and answer.
Thank you, Caitlin. Can you hear me? Can you see my slides?
Yes.
Yes.
Yes, cool. Thank you. So it's a pleasure for me to be here. After the presentation from Tommaso, you have a better idea what PHOENIX it is. And this presentation is mainly focusing a very brief description. But as Caitlin said, after that, we have the roundtable and you can make as many questions as you wish. So the size of the nanoparticles used in nanomedicine is what makes nanopharmaceuticals different to other medicines. And this affects the role of the R&D&I sector behind the mother biotech and pharma companies. This is one of the most important key differentiator feature of this technology. And in fact, the reduction of the particle size gives rise to specific properties like physical, chemical, and biological characteristics that differentiate the in vivo behavior of nanopharmaceuticals and compare to conventional chemicals. And these feature characteristics can lead to improvements in the efficacy when comparing a nanomedicine with a traditional drug. It's important to understand how the particle size affects the properties of the nanomedicine itself and the overall process that a given nanomedicine needs to undergo until reaching the clinics and the market. However, despite the efforts done during the last years in the field of nanomedicine, there are still difficulties on moving from one state, from the bench, to the other, to the market, mainly due to the problems in repeatability to obtain high quality and quantity products in GMP, as Tommaso mentioned in the first presentation. PHOENIX-OITB is a unified body and an operational structure to coordinate the value chains that merges of services necessary to develop nanopharmaceutical solutions and products to the end users. With this aim, our mission is to offer a single entry point for a consolidated network of different facilities, technologies, and experts for all different technology transfer aspects, from characterization, testing, verification, scaling up, GMP compliant manufacturing, or regulatory guidance along the long time of the development of a nanomedicine. As Tommaso mentioned, our service portfolio is divided into 5 different categories and altogether covers the different topics needed for the development of nanopharmaceuticals for early stage to enter into clinical trials. The first category is focused in accurate physicochemical characterization. The second category is including different in vitro characterizations. The third one in vivo. After that, manufacturing and scaling up, and finally, some innovation. Just going one by one, the first category is focused in the physicochemical characterization of nanoparticles, small and large molecules and biomolecules, and also the including of these molecules into nanoformulations. This step is essential to understand the behavior of nanomedicine and to provide guidance for the process of design, development, control, and safety assessment. In the case of nanomedicine, an adequate and complete characterization is in fact very important because the heterogeneity and the structural complexity of these materials cannot be measured just by traditional analytical techniques. PHOENIX brings together a wide palette of different analytical techniques that cover all different kinds of physicochemical parameters that need to be determined, and moreover, the knowledge of the physicochemical characterization of the service providers offer the possibility to develop new methodologies ad hoc to the specific problems that you can have under the development of these nanomaterials and nanomedicines. Here you have just a brief description of the features that we can characterize. Here there is also a table where you can see some of the techniques that we can develop, going from the R&D, GLP-like, GLP and GMP. We have around 12 different attributes that we can measure and more than 50 different techniques and assays that we can carry out. In the category second and in the second and the third category that they are focusing the in vitro and in vivo characterizations, the preclinical pharmacology or nanotoxicology compiles a set of crucial parameters that needs to be determined in order to successfully translate innovation and medicine into clinics. The absence of a specific regulation framework for nanomedicine and the lack of specific bioanalysis protocols for the in vitro and in vivo characterization of nanomedicine hinders and slows down the progress of translation of them into clinics. In PHOENIX, we count with a large number of different texts between these two different techniques, going from composition, bioactivity, immunoresponse, toxicity, cell viability, and so on. The tests provided for these in vitro and in vivo characterizations are, most of them performed following OECD and ICH guidelines. Here again, you have a short list of some of the techniques that we have developed. We have more than 70 different techniques to measure around 22 different attributes for the in vitro characterization and also for the in vivo characterization, where we can measure around 6 different attributes and with more than 14 different techniques and assays that we have currently in our service portfolio. In the fourth category, that probably is the most unique that we have in our OITB, because in fact our OITB was focused mainly in the scaling up and manufacturing, and this is one of the most challenging steps in the nanopharmaceutical development. The scaling up and the reproducibility of the production process of nanomedicine is tricky and is very difficult to reach. And this is very important to be able to achieve the industrial manufacturing stage of nanomedicine. In order to do that, we need to solve some issues like the lack of batch-to-batch reproducibility during the process of manufacturing, the long-term stability of the product, so the complexity of the manufacturing process, and also ensure sterile conditions of the materials that they are produced. In PHOENIX-OITB, we offer to our customers GMP and non-GMP manufacturing and quality control for the development and manufacturing of different kinds of nanoparticles formulation. And in fact, it is almost impossible to offer a one-size-fits-all testing, scaling, and manufacturing platform. However, since the challenges of scalability and sustainability and GMP compliance confront all nanopharmaceuticals and their preparation methods, a similar strategy can be employed for reducing the risk and overcoming the barrier to market penetration. At the end, we have a learning process going from the experimentation and the data analysis during the documentation, during the development, provide also the estimation of translational burden, monitoring during the scale-up provide a preliminary indication of process performance, and the knowledge obtained along the whole tech transfer and scaling up process is important also to translate it into a control strategy. In PHOENIX, PHOENIX-OITB concept, implementation strategy for the design in order to cover a wide range of nanopharmaceutical manufacturing methods, formulation administration routes, and also we count with excellent facilities, EMP facilities, that they are dedicated for the production and for the manufacturing of different kinds of nanoformulations. The last category of services that we have in our portfolio is related to the innovation. And in fact, in this category, we include several transversal services that helps throughout the whole process of developing a novel nanomedicine from bench to clinics and into the market, including such as quality by design, that this is necessary to identify the characteristics that are critical to quality from the patient's perspective, to translate them into a product, the clinical quality attributes, and establish relationships between variables of manufacturing. We provide support through the customers that they are not used to this concept of quality by design. We can also provide advices by design of safe and sustainability by design. And also we have some services related with training in order to support the rapid advances in the nanomedicine field. We provide a quite wide range of services that draw on its long-standing expertise in nanopharmaceutics, testing, safety, sustainability, and quality control, as well as in the latest regulation updates on nanomedicine field. And also included in this last category, we have our regulatory support. And in fact, the lack of agreed regulations for nanomedicine, that makes it difficult for researchers and SMEs to translate to clinics their developments. And in PHOENIX-OITB provides support and guidance to our customers for the creation of different IMPD of the nanomedicine under development, for different kind of products, different therapeutical areas, for EMA, for FDA, at different stages and with different models. At the end, I would like to highlight that this the service portfolio that I have briefly shown you here is available in electronic format in this link that I show you here. A summary: our service portfolio pretends to cover all the needs from research groups, SMEs, or large company in order to bring their products earlier in the market. And we are a non-profit, self-sustained, independent legal entity, and your direct contact with PHOENIX SEP is Nasende Gundai, that is our project manager. And thank you for your kind attention.
Okay, thank you so much, Jesus, for giving us this overview of the services. And we look forward, after a short break, to come back with service providers from each of these 5 areas for some questions and answers. So we'll be gathering back now with our speakers for our roundtable. Here we have on the screen, you heard already from our coordinator, Tommaso Serchi from Luxembourg Institute of Science and Technology. He's going to be covering the aspects of in vitro characterization, and of course also general questions. We have Jesus, who you heard from as well, from CSIC. He's talking about the physical chemical characterization. We have, in addition, some new speakers now. We have Ivana Vinković Vrček from the Institute for Medical Research and Occupational Health in Zagreb, Croatia. She's talking about in vivo characterization today. We have Alba Cordova from Nanomol Technologies in Barcelona, talking about CMC, chemistry, manufacturing, and control. And finally, we have my own colleague from BioNanoNet. We have Daniel Garcia. He's our regulatory expert, so covering innovation and regulatory support. Who could not be here today is our scientific coordinator, Nasende Gundai Tirelli from MyBiotech. She's also the CEO of the Single Entry Point, so the company that's coming out of this project. We'll have her contact info again at the end, so she'll be your first point of contact for any questions about the Single Entry Point services or accessing the services from the PHOENIX-OITB. So I'll take the slides off the screen now. so that you can focus on the speakers. So we've prepared some questions, and please at any time you can put questions in the chat and we will add them when we get there. So I want to talk to Jesus for just a really general question. You touched on this a bit in your presentation, but what are the challenges that are facing companies that are using nanopharmaceuticals in particular that's really unique to this field that PHOENIX can be addressing?
Yeah, that's a very interesting question, a very good question, because this is the origin of our project. I mean, the main issue is that if these companies are not familiar, they are pharma companies or small companies developing pharmaceuticals, that they are not familiar with nanotechnology, with nanoparticles. In general, they don't know how to handle these nanoformulations of these nanoparticles. And it's not easy because it's not the same kind of characterization that needs to be done for these nanoparticles as for conventional chemicals. If it is in the other way around, that there are groups or SMEs that they work on nanotechnology and they would like to jump to nanopharmaceuticals, in general, they are not used with the requirements of pharmaceuticals. And also this combination between nanoparticles and active principal ingredients is not easy, and they need extra support in order to develop these kind of products and to bring them to the market.
Okay, thanks a lot. I apologize if you hear some church bells in the background coming from next to my office, but hope you can hear the next question. So Jesus, I'll stick with you. So you're also one of our experts for physical chemical characterization. And that reminds me to say to the audience, so these are not our only partners and our only experts. So we have several other service providers. We just focused on one from each of these categories to keep it simple today. But staying on physical chemical characterization, are there unique tests or unique tools that you need to perform a high quality characterization of your product so that you can avoid any inaccuracies later on as you scale up?
Yeah, I mean, the main strength that we have, as you mentioned, we have different partners and we have different experts and we have different facilities and infrastructure. And this is one of our strengths, that we can scope a really high scope of different techniques and different characterization techniques, not only PhysChem, also in vitro and in vivo, but Tommaso and Ivana can explain more about that. But in the case of PhysChem characterization, in some cases we need some big facilities like electron microscopy, or sometimes the techniques and the methods that needs to be developed ad hoc, they are not so complex, and they are just PhysChem techniques that need to be adapted for nanopharmaceuticals, for nanoparticles. But you need to know and to understand the nanoparticles and the behavior of these nanoparticles in order to develop these ad hoc techniques. In general, what we have in our group, so also with the customers that we have in PHOENIX, or we can have in PHOENIX, is in general they come to us because they cannot develop on a specific technique because they don't have the facilities or they don't have the experts to run those infrastructures like electron microscopy, atomic force microscopy, for example, and so on, and they need this data for their development. Or in some cases, they come with a very specific problem that they found when they went to the FDA or the EMA. They request them for a very specific question, and they need the answer, and they didn't know how to answer that. They didn't know how to develop a specific physicochemical technique to answer that specific question, and that is why having a large group of experts in the field made that very strong, because I mean, we can have different expertise, more in microscopy, more in analytical techniques, more in vitro, in vivo techniques, and at the end we can provide with an answer to this specific question.
Great, thanks a lot. I'll move for the next question to Tommaso. Tommaso, in your overview presentation, you talked about this innovation valley of death. I think a lot of people are familiar with. And you're also representing the in vitro characterization today. So at the stage of in vitro characterization, this valley of death is often due to bad study design because of a lack of knowledge of how to really handle nanoparticles. So can you tell us, Tommaso, a little bit about your expertise with nano, and especially if you're comparing services of other CDMOs out there?
Yeah, sure. We do have a lot of experience in nano, not necessarily only in nanopharmaceuticals. We do a lot of different toxicity studies using different nanos. We have used, you name it, silver, silica, carbon nanotubes, lipids, whatever. So many things can be motivated from chemical testing because the principle at the base of testing is conceptually the same. So you want to see an effect. If it is a therapeutic effect, you will measure things in a certain way. If you want to see toxicity, you will measure it in a different way. But the concepts are similar to those of chemicals. However, there are a number of different assumptions, of assumptions which are different. And they have to be different due to the intrinsic nature of the nanoparticles. One problem that we face, which is a bit simpler when dealing with chemicals, is to track the particles once you put them in biological system. Be it in vitro or in vivo, doesn't really matter. Most of the particles that we use in the nanopharmaceutical sector are carbon-based, poorly carbon-based, which means that for most of the technique that we use, they're invisible. Which means that when you do the study design, there are mainly two main things that you want to see. Where it goes, where it goes, where the chemical which is loaded goes when it is released, the dynamic of the release, and so on. And what you can do, because basically you can measure it as it was a conventional chemical, but you lose completely track of the particle. So there is a tremendous push at an international level to develop and validate and harmonize methods to do it, and we're simply not there. So one big problem is in this. So when the company is asking us, can you tell us where it goes? It depends on the particle. Most of the time, when it is the kind of particles that we normally use carbon-based, we cannot. And this is a major problem. Then yes, as I said, there are some things which have to be adapted. For many endpoints in toxicity, we don't need to do too much. But there are some special cases when you want to do inhalation studies, when you have to take in account agglomeration of the particles, how you disperse the particle, if it is a dry material, if it is not a dry material, and so on. Things which you also have to take in consideration for chemicals, but there we have more knowledge. So we started hundreds of years ago to test chemicals, while the nano field is relatively new. So we are trying to do the same things that we do with chemicals, but with only 10, 20, 30 years of experience. So we will reach the same point, and this will also help to overcome some of the issues in the development, but we still need some time to develop new techniques and to harmonize what we have already now.
Okay, thanks, Tommaso. Ivana, I want to move now to you. Ivana is our expert for in vivo testing, and I want to ask you about critical quality attributes. So can you tell us a little bit about them, and what is the minimum information related to the critical quality attributes that you have to provide, or that a customer has to provide before starting in vivo testing, and how can PHOENIX help them with that?
Yeah, thank you, Caitlin. Rather to answer directly to this question, because it's a very broad and very long answer, I would rather highlight the advantage of PHOENIX-CEP regarding the determination of the minimum critical quality attributes for later preclinical and clinical testing. The PHOENIX-CEP in fact involves complementary expertise of different service providers. So here you can see Tommaso, Jesus, Daniel, myself. So we are very complementary with a long expertise and a broad expertise of dealing not only with nanopharmaceuticals, but also with different types of nanomaterials. And all so lipid-based nanoparticles, peptide-based, metallic nanoparticles, etc. So what is known for all who work with nanomaterials, it's that nanomaterials have very peculiar properties, depending also on how they are designed, and it's not possible to provide single technique for each type. And this is why PHOENIX-CEP established service providers from different fields with different facilities. And for my service, so from my point of view, I provide services for in vitro and in vivo characterization, biocompatibility, and this PHOENIX project facilitated our decision to establish GLP laboratory. So we are now in process of getting a GLP certificate for our laboratories, which will be fully complementary to work with nanomaterials. And this is something that is unique in PHOENIX-CEP, because all of us are experts for nanomaterials, and we really can and are able to help people that want to develop and to design the nano-enabled medical products, either medical devices or nano drugs. So this is something unique, and it's already recognized by representatives of Nano-Industrial Association, that there are no such CROs and laboratories with certificates that are fully devoted to nanomaterials, and we are fully devoted to nanomaterials.
Okay, thank you for that answer. If that was a short answer, then the long answer would take us a bit while. Okay, I'm gonna go to Alba now. Alba Cordoba from Nanomal Technologies, talking about CMC. Alba, everybody is talking about getting to a GMP compliant environment for manufacturing. What are the actions that end users can introduce to their nanopharmaceuticals R&D phase to make sure that they have a smooth transfer to GMP compliant environment?
Yes, thank you, Caitlin. I think that this is like a complex question because it needs to take into account several factors, because as mentioned here, well, pharmaceuticals are highly regulated. So if you are coming from an academic environment, the first thing that you need to take into account is like the overall picture of drug development cycle, and also see where are you heading to. So I think that the most important thing is to envision which kind of nanopharmaceutical are you going to manufacture, so have very clear a target product profile since the beginning. Maybe Daniel will give more insights about this point later, and then define the administration route. Will define also which excipients will you need to use, and which excipients then you would need to check, for example, if they are available in pharmaceutical grade, because this is required for GMP production. And also you need to check if you have suppliers for this excipient and also for your API in this pharma grade. This would be like the first steps. But then I think the main thing would be to define as early as possible the manufacturing process. I think that this is like a key point, because to ensure the smooth transfer, the manufacturing process can have a great impact, because most nanopharmaceuticals and nanomedicines are defined, the product is defined by the process. So changing the process at the later stage can have like a huge impact that maybe can stop the development. So I think that I would first of all think also in the scale that you will need at the end. So which manufacturing scale will be needed for the clinical stage, for the industrial scale, and which manufacturing method can be applied at GMP level to achieve these amounts and quality. So I think that this would be like the key points. Also, I think it is very useful to since the beginning apply a quality by design approach that has been introduced before by Jesus. That this is one of the services offered by PHOENIX, and it really helps to ensure that you're having a robust manufacturing process that is giving you a robust and reproducible quality of the drug product, so that you are controlling your specifications, that you are having a well-defined list of critical quality attributes, that you know how your process is impacting your quality and how your materials are impacting your quality. So definitely quality by design also can ensure a smooth transfer. And I think that last but not least, it would be very important to check the costs as soon as possible, to be aware of which is the amount of time and the cost that your product will have in, for example, for having a clinical test material, and also the clinics itself, and also like the whole development, and to see which, for example, which alternative is feasible and which not, and also just to keep it simple. So keep the method simple, keep the product as simple as possible, engage with the regulatory authorities, and also seek advice from the experts that can help you. And also, just to finalize, I think that it's important, for example, to check for IP issues as early as possible to ensure that you have freedom to operate, so that you are not developing a product that later will not be able to be marketed. I think that's the main general points.
Thank you. And I think that these are all aspects that PHOENIX partners can help the customers with. And speaking of that, so Alba, when is the right time for a customer to take action for scale up, and how can PHOENIX help companies that might not know exactly what to do when along this process?
Yeah, so it's important to think in scale up as early as possible. So as soon as you have a preclinical lead with some efficacy results, it's important to define which manufacturing process will be used to ensure that you can reproduce the quality for all the preclinical testing. And also sometimes scale up is challenging, and more for pharmaceuticals that require, for example, new and innovative particle manufacturing technologies, so that are not yet like in the state of the art in the pharmaceutical manufacturing industry. But well, I think that luckily now with all the COVID vaccines manufacturing, we are like speeding up all these manufacturing methods. So as early as possible, define the manufacturing process, and also try to anticipate the requirements, for example, for the clinical phase. Try to design as early as possible which amounts will be needed, and also for example for the GLP package, that sometimes more quantity is needed for all the preclinical regulatory, for example, toxicology and with second species and so on. Sometimes the amount is bigger than for the first clinical trial. So you need to ensure that you will have the same reproducible product in all the subsequent steps. So scale up must be clearly taken into account as early as possible.
Thank you so much for that answer. Danielle, I want to go to you. Danielle from BioNanoNet, giving us regulatory advice. Getting advice from EMA, from the European Medicines Agency, it's quite challenging for some companies. Are there any kind of roadmaps or materials that companies can use during nanopharmaceutical development that can help them prepare for talks with EMA or with the FDA? And how can PHOENIX help them here?
Yeah, thanks, Caitlin. Getting advice from EMA, especially for nanopharmaceuticals, is an option, of course. I mean, there is basically two main tools in Europe, no? Especially we talk about the European Medicine Agency, which is the service they provide through the Innovation Office, what they call ITF. This is for early stage of development, so sponsors can go there and have what they call informal discussions, no? Where they will bring kind of a briefing document with some key questions, no? And later in time, when you have progress, normally you can go for scientific advice. So in principle, sponsors can get advice from EMA either at early stage or along the whole development, because you can request scientific advice at any time. And the EMA is obliged to provide you with advice, as long as you follow the procedure and as long as what you are intending to discuss is appropriate. Let's say it's not describing guidelines. So everything is describing guidelines, should not be discussed in a scientific advice, no? So basically, what they're gonna need, sponsors are gonna need kind of a document where they explain their developments, and that's key that these kind of documents they are easy to read and they contain the appropriate details to promote the discussion, no? So of course, regulatory expert can help you in putting some architecture, no, in these documents, some kind of flow, making sure that all the key elements are there, and to promote a positive discussion, no? The main limitations for, especially for SMEs when they approach regulators is, well, sometimes the inexperience make them to feel uncomfortable in the discussions, or even to don't provide like a position, because this is also key, no? The sponsors need to understand that most of the time when you approach a regulator, it is expected that you have a position as a sponsor, and the discussion is based on your position, no? And as I say, I mean, of course, a regulatory expert can help you in selecting the right place or the right agency. Either you go to central or you can go to national agencies, depending what you are trying to solve. The right time also to have like a strategic approach to the regulators, no? And for making sure that the all elements are eventually reflected in a formal letter, no? This is more or less the point, no?
Okay, thank you, Danielle. I think that the regulatory questions give people a lot of uncertainty, so it's really good to know that they have a place to go for specific questions. And of course, these all depend on the individual situation. But still, is there a minimum preclinical package to generate enough possibility for regulators to accept starting first in human studies? And how can you guide a company here?
This is a key element, especially for research jumping into more like into the clinical field, no? Small companies that came from the research area, and then at some point in time, because they have some valuable leads, they decide to progress, and they have the funding, no? And it's also always a key questions whether, I mean, I have enough information. In general, my experience is that, especially people coming from the research side, they tend to produce in certain fields, for example, they are marked into efficacy rather than into safety talk. So one of the main mistakes I notice is that mainly from small biotechs coming from the research area, they tend to be more focused on efficacy rather than safety and toxicology. So one of the points is also to focus also on the other element, which is both of them will make the right balance to jump into clinical. And then in general, basically, you need positive data in terms of efficacy. You need positive data in relevant animals of the disease. So the relevant models, what you have obtained positive data. Whether you need more than one model really depends on how uncertain has been the translation in this area of the disease. Of course, sometimes regulators will accept a single model, whereas the sponsors or the people who is putting the money will like to reduce the cost and will like to see more than one data coming from more than one model. No? And on the safety toxicology side, the plans is also something we can help, but need to be adapted case by case. And of course, the idea will be to produce the minimum data to cover the preliminary clinical study. So I mean that at preclinical level, we produce safety tox data to cover initially the phase 1, the escalation, even the proof of concept. And when into the clinical data, we continue and produce the rest of the tox data. No? There are key elements, for example, for research. They have a lot of questions regarding the level of characterization of the mechanism of action before you go to humans. Because this is important because you don't need to characterize the mechanism of action, the entire mechanism of action. No? The level of detail you need to jump into humans will really depend on a case-by-case basis, whether you are assuming more or less risk. But again, this is also something I see commonly, that is people coming from the research side tend to go much into detail. Sometimes it's too much detail for a mechanism of action and just leave safety talks aside. So you need to focus on both. This is more or less the overview. Okay, Caitlin?
Yeah, thank you so much. And of course, everybody can go for specific questions then to the single entry point. I want to turn on to the audience to see if anybody in the audience has additional questions for our speakers. They can be specific, they can be general. You're welcome to write them in the chat. You can also, I think you can unmute your microphones as a participant. We can find out if you'd like to just ask directly. And we'll just give you a second to see. And otherwise, I put in the chat a link to the website for the single entry points and newsletter. So if it's sort of processing the initial information now and you have more questions in the next couple weeks and months, we're happy to take them. We know that this is a complex field and the questions are very individual. If there are no questions at the moment, and please keep them coming if there are. Jesus, I want to come back to you for a final one from my side. So if I'm a company and I think I might be able to benefit from the services of PHOENIX, but I'm not really sure, what do I do? What are the steps? And what would it look like if I were to take advantage of these services?
Yeah. So the first thing is to contact to the PHOENIX SEP with the link that you put in the chat. Just go to our webpage. There is the link of our contact person, that is our CEO, that is Nascende. And immediately after, we will establish an interview with you in order to see your needs, also to share with you our capabilities. After that, we will have an internal meeting in order to see what we can propose in order to solve your questions and your needs. And we will end it with a proposal and also a budget that we will propose to the client. And finally, once everything is agreed and everything is established, we will start to work on the project that you propose. The first thing is the contact point for the PHOENIX SEP that you can find it in the webpage.
Great, that sounds very easy, I think, for our customers. All right. Well, I'd like to thank everybody from our roundtable for the questions and the thoughtful answers. Of course, as they say, we're always happy to hear more as we are growing and developing the company. I'm going to share the last contact information here. So as we explained before, PHOENIX-OITB was a project, or is still a project, running another year or so. And out of this project, we created a company, this single entry point. So this is why we have the two different websites. So you can look at the project website. That was the first link that I shared. And then also the phoenix-sep.com for the single entry point. But you can reach us either way. Don't worry. We'll get your question in the right place. Here, I'll just share the slide again that Jesus had before. So the single entry point is this legal entity. Your direct contact here is Dr. Nazende Gunde Tirelli. You can scan her contact information here or find it through the website. And again, you can have this initial meeting to see what your options are, kind of mix and match the services and get a plan created. And we'd love for you to stay in touch. We have a link to the newsletter. We have one for the project and for the single entry point. The project will end, but the single entry point will be, and the websites and newsletters will be continuing, of course. But we'd love for you to follow us on LinkedIn. This is where you can also get the latest news, and you can also see when we'll be at different conferences, mostly in Europe, but not only. So please connect with us there for the PHOENIX-OITB for the project and the PHOENIX-SEP for the single entry point. So thank you everybody for coming and for your attention, and we look forward to being in touch in the future. All right, have a great day everybody, and thanks again to my speakers and to the participants.
Thank you, Caitlin.
Thank you.
Thank you, Caitlin.
Bye-bye.
Thank
We'll
you.
see you at the next events. Bye-bye.